I think we can get started then. Welcome everyone. Thanks for joining us today. We have Professor Caroline Uhler from MIT in the Broad Institute joining us, and Caroline works on problems at the intersection of machine learning, statistics, and genomics, with a particular focus on causal inference. She's been at MIT since 2015. Before that, she was at IST Austria and got her PhD at UC Berkeley in statistics. Her research has been recognized with numerous awards and fellowships, amongst them Sloan Research Fellow and Simon's Investigator, just to name two of quite a few. So in any case, I'm really happy to have Caroline here to tell us about her group's work on causality and autoencoders. Thanks.
Thank you so much, and thanks for the invitation. Yeah, so I'll tell you a little bit about what we've been thinking about at this intersection between, causality and autoencoders, and then how this relates to different kinds of biological questions that we've been really excited about. And I'll start with a motivation slide, mainly coming from the fact that, we started a new center at the Broad Institute at this intersection between the biomedical sciences and machine learning, and spent quite a lot of time thinking about, what would be the one machine learning area that might have, that would really propel questions in the biomedical field, but also the other way around, where the biomedical field might actually lead to very new questions in that area in machine learning. And so for us, this has been, or we got to the conclusion that this would be something like causal representation learning, that would be really important to develop in order to answer many different questions in the biomedical sciences. So why causal representation learning, and what do I even mean by something like that? So if we think about the biomedical field, we have huge amounts of different kinds of data modalities at different scales. This can be from gene expression data to, all kinds of different imaging modalities, be it at the cell level or at even the whole, organ or body level. All of these are in very different data modalities. Often we don't know the kinds of questions that we're asking, right? We're looking for new biology. That means we have unlabeled data because we don't even know what to label. And so it is pretty clear that in this setting, when you have, huge amounts of very different data modalities and unlabeled data, representation learning has, of course, been, hugely successful in areas where, say we know from vision or integrating text and vision, etc., for different kinds of modalities where, these kinds of methods have been really used and very, very successfully used. So why causality? And this maybe comes more to, in terms of the biological questions that we want to ask. Well, so if you think about the more standard applications of machine learning, where machine learning has been really successful, let's say, for example, a recommender system. Now, in a recommender system, you really don't care about the underlying mechanisms, right? In a recommender system, if you know that someone bought ice cream, say, then, it's sufficient that you can also predict that this person might also be interested in ice cream, but you don't really care about why this is the case. You just care about, high accuracies in terms of prediction. And this is what, of course, machine learning algorithms are mostly trained to do. Now, if you look at different kinds of questions in the biomedical field, well, there you really care about the underlying mechanisms. Here is a very well-known data set of melanoma, where the task is to be able to identify benign versus malignant melanoma. And it just so turns out that in this particular data set, which has been used in many, many different studies, and it turns out that the benign class is overrepresented for these bandages, whereas the malignant class is in fact overrepresented for these measuring bars. Now, if you just train, say, your standard neural network on this task, then this is one of the important features that it is going to use, because it will just use whatever you give it, right, in order to achieve high accuracy or prediction accuracy. And of course, if you then apply something like this in a new or different data set or a different hospital setting, well, then you're going to do really, really poorly. And the reason is because these features are just not causal, right? If you were actually using some causal features about the melanoma itself, well, then you could translate to a new hospital setting. So we really want to get at the underlying mechanisms, not just for actually doing well in different kinds of scenarios, but also to actually understand the underlying biology. So this, to me, leads to the question of, how can we do representation learning, but to actually identify representations that tell you something about the underlying mechanisms. So something like causal representations, and I'll talk more about what I mean by this. And of course, this will be difficult, right? So, for other applications like recommender systems, well, maybe it would also be useful to have causal representations. So what makes me think that maybe in the biomedical setting, we will be able to do something like this that is very different than in many other machine learning applications. Well, in the biomedical sciences, we have this huge hammer, which is given by perturbations. So it is quite easy to perturb things in the biomedical sciences. You can do knockouts, for example, right? We'll know of CRISPR, so we can knock out any gene we like. We can apply drugs. That's another perturbation, right? A drug will only have an effect, it's causal. A drug will only have an effect on something that is downstream of the targets of the drug. We can perform mechanical perturbations to cells, et cetera, So we have this huge hammer, which is actually perturbing the system. And that, of course, gives us information about causality, because it will only have an effect on something that actually comes downstream of it. So I think perturbations are something that is quite unique in the sense that, in this large scale of what we have access to in the biomedical sciences, and could help us to get at causal representations. Maybe even to step so this will be at the large picture. What I would like to do is develop something like causal representation learning. Just to have a couple more particular biological problems in mind that we've been working on quite a bit. I have here a slide that will also make it clear again why we need causal representation learning. So a lot of important problems in biology could really be put into this framework, into this transport problem framework in single cell biology. And let me just go through some of these examples, and then it will be quite clear that for all of these, how I have represented them here, they will actually lead to causal representation learning questions. So maybe just very basic question of, you have all of these different kinds of data modalities, say in the single cell field, we would have different kinds of imaging data sets. So here, single cell images. So every one of these is a cell. This is just a cell nucleus that you see here that is colored. And at the single cell level, also another data modality that is very abundant is expression data, where again here every row is a cell, and it has like 20,000 entries, and that's just the expression of every one of our genes. So now the problem with many modalities in biology is that they are destructive to the cells. So if I take this measurement here of the expression of all of our genes in this particular cell, then the cell is dead. So that means I cannot also get an image. And the other way around often as well, in order to get this image, I need to fix the cell, and thus then the cell is dead, and I cannot also get the expression output. So there are really important questions of how can I actually translate between different data modalities or integrate different kinds of data modalities? In particular, you would like to go from cheap modalities to, more expensive modalities. So from imaging, say, for example, to expression, without ever being able to measure these modalities in the same cell. So these are kinds of questions about transporting here between different data modalities, or transporting and integrating different data modalities. Here they're at the same scale, but what we saw on the previous slide, right? They can also be at different scales, where you have some data at the cell levels, and then some data at the tissue level, et cetera, that you need to integrate between. Different problem. So here clearly, right, a representation learning question. I'll get to why this is maybe also a causal question on one of the next slides. Similar problem again, like before, but maybe I don't have different data modalities, but I often have different time points. So say during cancer progression, I'm looking at, one time point and then the next time point. I cannot take a movie because these processes that we care about often take too long, that you can actually not follow the cells over these many years, say, for example. So you would like to, you have sometimes some data set taken at time point 1, some data set taken at time point 2. So the question is, how can I transport between different time points? So predict how these cells would look like at a different time point, forward or backward in time. Very similar question to this one, of course. I can just have, instead of data modalities, I can just have different time points. So that's all at the observational level. Now, if we get to more of a causal kinds of questions, where I actually have perturbations, as I said, we do have often access to very large scale perturbational data sets. So here I can ask, again, different types of questions. So this is the question here that is, behind basically all of pharma industry, right? Is like, I have my drugs and I have tested them on mice. And now I would like to predict what this particular drug does in humans. Right. So instead of like asking it in this extreme way of transporting from mice to humans, I can ask it in a different way. And this is the question that I will be asking, that I will be working on also in this talk, is like, let me actually just translate or transport from one disease context to another. So say you have done a large drug screen in cancer, in different types of cancer cell lines. Now here comes a new disease, and you care now suddenly about SARS-CoV-2 infected lung epithelial cells. Well, I've already done this large-scale drug trial on all these different cancer cell lines. Can I from there predict what all these different drugs will do in this new context, namely on SARS-CoV-2 infected lung epithelial cells? And if I can do this prediction, well, then I can rank all the drugs, right? Because what I would like to do is actually choose the drug that pushes me back from the infected state, so from the SARS-CoV-2 infected lung epithelial cell to the normal lung epithelial cell state. Right, so I would like to just predict the effect of all these drugs in this new context, and then just choose the one that is most anti-correlated with the signature of the disease. So this is, of course, a causal question, again, because a drug is an intervention in the system and only has an effect downstream. So this problem is known as the causal transportability problem of transporting the effect of a perturbation across different contexts. I can also ask the question differently. So instead of transporting across different contexts, like different cell types, different disease contexts, different organisms, I can ask a question of, like I have the space of different kinds of perturbations so big in biology that I'll never be able to just test them all. Say you're looking at knockout data, right? We have 20,000 genes. So yes, you can go in nowadays and actually knock out one gene after the other. But, then we care about combinations of genes. So you'll never be able to do all combinations of even, say, 5 genes out of 20,000. And then you want to do this in all kinds of different cell types. So, or, you have some molecules that you're testing, but the space of all molecules is so huge that you'll never be able to test all molecules. So you need a way of transporting between different interventions, right? So I have tested some interventions, either molecules or genetic perturbations in, say one cell type here. These are all of my observations. And now I would like to be able to predict from these. I would like to predict, well, what will happen if I do a new perturbation? Right? Can I predict what these particular outputs here would be? So this is another transport problem, again, a causal transport problem, where I'm actually going not between different contexts, but actually between different kinds of interventions. So clear here that I have like some kind of mix of representation learning and some kind of causality, right? Which I need to find good representations in order to actually be able to do these kinds of prediction problems. Okay, so these are the questions that will motivate much of what I'm going to present in this talk. And so what I wanted to do is just give, first tell you a little bit about how these questions have maybe been looked at for a long time now, and then give you a little bit of some new ideas of how I think maybe we could get there to actually tackle some of these questions. Okay, so, traditionally how these questions have been looked at trying to really predict the effect of a perturbation is to really go and understand the whole gene regulatory network. Okay, so this is kind of the gold standard of what people have been wanting to do, because, right, if I can actually learn the full underlying gene regulatory network, well, then you can, really predict what will happen if I go in and just intervene on this node, or if I go in and intervene on this node, or even on combinations of nodes. Right. So this is really what has been like kind of the general approach, is just to really try to go in and understand the full underlying gene regulatory network. And one, and this is like the kind of framework that I have been using in my research, is this framework of causal structural equation models. So the whole, kind of basis for how to think of or how to even try to learn such gene regulatory networks has been set up by actually someone who comes from the biological field and has worked a lot in statistics, by Sewell Wright, who has thought a lot about inheritance or heritability of different traits. So these directed graphs that you see here are representing, inheritance of different traits, and he really set up the framework for thinking about these causal structural equation models. So here, every node is a random variable. So that would be, in this genomic setting, that would be a gene. So here, X1 would be a gene, X2 would be a gene, X3, etc. And then these directed edges, they correspond to causal effects. So here, for example, and the model underlying it would be, every node is just some function, can be highly nonlinear, of its parents and some noise. Okay, so here, for example, X1 is a function of just its parents, so that's X3 and some noise. And this can be any function, and noise can be any noise, can be Gaussian, but of course also anything else. So this is the representation of how you can think of here, for example, by representing a gene regulatory network by these causal structural equation models, or also how Sewell Wright got at this by representing, inheritance of different kinds of traits using these causal structural equation models. Okay. So now, of course, what we want to do is to learn a graph like this, right? So you want to learn these underlying gene regulatory network. Nobody tells you this underlying gene regulatory network, right? I would like to learn this just from data on the nodes. So I get to see the expression of all of these different nodes, and I would like to learn this kind of data. And, I thought at the time that we have, these data sets in genomics have become so big that you have many more samples than the number of variables, although the number of variables is like 20,000, right? Humans have about 20,000 genes. Nowadays, many of the data sets, even if you just do one experiment, you get 100,000 cells at once. So, these are really quite large data sets. And so you could think that maybe, nowadays the time is ripe that we can just from such observational data actually go in and learn these gene regulatory networks directly using, say, an underlying causal structural equation model. Okay, so what are the methods that have been developed for actually learning, from data on the nodes, learning these causal graphs? So here there has been really amazing work by various people, right, on this really hard problem of how do you actually get a causal relationships from observational data. In particular, this problem of learning the underlying graph was spearheaded by Spertus in the 1990s. And here I just want to show you one little bit of work that we've done, very quite early on, which really kind of shows how hard this problem is, and something that really will come up when you go from more theoretical work like what Spertus has done to trying to actually use these kinds of algorithms in practice. And so here is an assumption that is a fundamental assumption when you want to actually learn these causal graphs. It's known as the faithfulness assumption. What this faithfulness assumption says is the following. It says that, say here we only have 3 genes instead of 20,000, right? So let's say we only have 3 genes, and, gene 1 has a positive effect of gene 2. So that means if gene 1 is up, the expression of gene 1 is up, then maybe the expression of gene 2 is also going to be up. But gene 1 down regulates gene 3, and gene 3 again upregulates gene 2. Okay, so now what the faithfulness assumption says is that these different paths, right? So if gene 1 goes up, then gene 2 goes up, but at the same time, if you go along this path here, gene 2 goes down, right? So it could be that, these 2 paths cancel each other out, right? But what the faithfulness assumption says is that these 2 paths are not allowed to cancel each other out. Because if these 2 paths cancel each other out, then you have a problem, because then in the data, you might observe that gene 1 and gene 2 are actually independent of each other. But if they're independent of each other, that means that I'm going to remove an edge in any of these algorithms that learn these causal graphs. So that would be a problem, right? Because that would not correspond to the true graph. So I can then, in that case, not learn the true graph. Now, of course, exact cancellation of these paths is very, very unlikely. In fact, it has probability 0, right? or measure zero, I guess, if you're looking at it in some high dimensional space, or in this case, in some three-dimensional space. But the problem, and so that's why it was always believed that this faithfulness assumption is maybe not so problematic. But when you work with data, right, then we have to do conditional independence tests. So we will be testing whether gene 1 and gene 2, the expression of these two genes, are independent or not. That means you have to introduce some cutoff where you're saying like, well, if the correlation, say, comes out to be smaller than this cutoff, then I'm going to conclude that they're in fact independent. Now, if you have to include a cutoff, well, then suddenly, this thing doesn't have measure zero anymore, right? So because even something small, I'm going to conclude that they're independent. And so suddenly, this faithfulness assumption, when you actually do it in the data setting, is not that harmless anymore. And in fact, what you can show is that this is just on 10-node graphs, right? And quite sparse graphs. So this is the sparsity over here. So here we just have, a density, so expected neighborhood size of 2. Then even if you have quite a lot of samples, or let's go maybe to density of 3, neighborhood size of 3, even if you have a lot of samples, like a 1 million samples, then, for sure, you will have a faithfulness violation, okay? Meaning that you are, in fact, going to learn the wrong graph, because you will always be somehow close to somewhere canceling out one of these different kinds of edges. So this was kind of the work that we've done, like, sometime early on, which then made me, at least, I was very hopeful to be able to use these kinds of algorithms for getting at, at these causal relationships from observational data and genomics, but is kind of discouraging to see, right? Because these are huge sample sizes with very small number of nodes. So if here you can prove results like this, well, then what will you do when you have like larger, you'll basically never have enough samples to run any of these algorithms on these very large 20,000 node graphs. However, and this is something that I mentioned at the beginning, what is exciting in biology is that there is interventional data, right? And so the question is, of course, well, if you have interventional data, maybe that gets you around this kind of problem that we have when we just have observational data. So maybe it is better. Maybe with interventional data, we can actually try to learn these causal graphs and then, learn these gene regulatory networks. So these are just to show like the interventional data sets that have been developed, for example, using Perturb-seq, where you can go in and really knock out any one of the genes you like and still get these very, very large data sets. So, and this has been some work that we have been very excited to do in recent years to really understand, when you have interventional data, how much does that rescue you? In some sense, how much can you then, use a combination of observational and interventional data in order to learn these causal graphs? And you can get to, consistent algorithms, meaning that if I have an infinite sample size, I can actually prove that it's going to learn the correct graphs. You still have assumptions like faithfulness, they're weaker than faithfulness. You can get to some results that, feel okay, are definitely better than random. And, it's not as extreme anymore as in this kind of setting. But well, are we happy with these kinds of results? With 24 nodes here with 15,000 samples, these are the kinds of results that we're getting in terms of number of false positives and number of true positives. Yes, better than just observational setting, but certainly not where we would like to be in terms of genomics. And this is just 24 nodes, right? Not on 20,000 nodes. And these algorithms, what you're gaining is really kind of, if you're, these, of course, when you want to have directions, right? How can you learn something, or what do perturbations give you in addition to observational data? It really tells you something about the direction of these edges. So because, if you perturb some node, like say in this case, gene 1, and you want to know something about the direction of these edges, well, if this is the true graph and you're perturbing gene 1, in this case, gene 2 and gene 3 would change, right? But if gene 1 was actually all the way downstream of gene 2 and gene 3, and you will perturb gene 1, well, then nothing happens to gene 2 and gene 3. So these perturbations are really informative about directions of edges. And so, it is clear that it should help you when you have perturbational data. The question is just how much does it help you, and does it really allow you to overcome some of these really fundamental kinds of problems when you're trying to actually learn these causal graphs? It is just a really hard problem.
Can I ask a question?
So, yes, please do. Yes.
Are these graphs acyclic? So is it really acyclic, or are there cycles in them? in reality?
Perfect questions. Yes. So there are the first algorithms that were developed all assume acyclicity. So there are algorithms for learning cyclic graphs as well, where you also do it based on conditional independence relations. I know Elena is here. She just sent me an email that they're working on generalizations of these types of algorithms that we have developed here for cyclic graphs. So you can do it's usually you pay a price, of course, in terms of computation time. But there are algorithms that can learn also cyclic graphs, and which is important if you're doing something for gene regulatory networks, of course, where you do expect there to be cycles.
Okay,
yep. Thank you. Yeah. So okay. So with all of this work, I think what one then what we got to then ask is, how much does a biologist? How much is it that that this is really our end goal of learning these kinds of graphs? And is it really that someone wants a graph on 20,000 nodes? And at least for me, when I have these discussions with biologists, the answer is usually yes. So this is really the end goal, and I do want to have this graph on 20,000 nodes and really understand it. But then if you probe a bit more and ask, wouldn't you maybe also be happy with, these kinds of problems? Like if I have a black box and you just ask me, and this black box can give out, like you tell me, like, I want to know the effect of, this kind of perturbation, this knockout. Would you be fine with having a black box that can predict the effect of any perturbation that you have not yet seen? Then usually the answer is also yes. Right? Because the question is always, what are you going to do afterwards once you have this graph? Is it that you're just going to use it to predict the effect of new perturbations that you haven't seen yet? And if yes, well, then this is a much easier problem, right? Should be a much easier problem. So I can solve, if I have like your perfect underlying causal graph, well, then I can solve this problem of predicting the effect of an unseen perturbation using that causal graph. But, maybe I can also solve this problem without knowing the causal graph or without fully knowing the causal graph, right? So this problem should be, or is hopefully easier than the other problem of going through, learning this full underlying causal graph. So then the question is, can we maybe just directly try to go here? Since, this is already gives rise to quite important problems biologically, right, that you care about, say, in the pharma industry, and might be easier than just directly going after understanding gene regulatory networks. And certainly give you some understanding of gene regulatory networks, since I can then predict the effect of any perturbation. Okay, so let me try to go a little bit in that direction. So you won't see anything about graphs anymore, but I want to kind of solve these problems without graphs. So let me go into a couple of, I just have 3 high-level ideas, and then I'll go deeper into one of these ideas. So 3 high-level ideas, and I'll start off with actually just making the problem a little bit harder first, because a lot of our data is not gene expression data. So gene expression data, you have data on every gene, right? And then I'm trying to learn these, say before, I was trying to learn these directed graphs among these genes, but at least I already have data on the genes. And we would think of the genes as being the causal units. Now, what about if I give you images? Well, the pixels are certainly not causal units, right? So if I give you images, then the first problem you actually have, even if you want to go this route of, going with learning a causal graph, is that you actually have to learn the causal units. You don't know what the nodes of the graph are, right? It could be that a particular protein is inside the cell nucleus or outside, or how much is inside, where is it in the cell? We just don't know. It could be any of these, could be the causal units. So the question already is, how do we actually learn causal units? And I think there is quite a lot. And, if you look at causal representation learning, this is another question that has been asked in this whole framework of, can we even learn what the causal units are if I don't give them to you in the first place? Even if you think of gene expression, maybe, genes are not really, they're usually seen as causal units, but it could also just be a full pathway that should be summarized as one causal unit. So how could you go about that? So that I think is like one of the most basic questions, and there's very little work and some nice work by Frederick Eberhardt. But here I'm just going to give like a very high level overview of some idea of like that actually relates to this kind of question of how do we integrate between different data modalities. Okay, so here is a framework, and it already gets to autoencoders that of course I will be talking quite a bit about. So first of all, autoencoders, I'm sure everyone here is familiar with them, but this is representing here an autoencoder where you have some input data set, say in this case, it would be images that go in. Then you usually have some neural network that maps you to some latent space. And we'll talk a little bit more about this latent space. And then you have another neural network that maps you back from the latent space back to the original, in this case, image space. And often these autoencoders are seen as some nonlinear version of PCA, so that they're used for dimension reduction. So you go from some high dimensional space to some small dimensional space. That's your representation of the data, and then you go back to your high dimensional space. Okay, so now what can you do if you have many different data modalities, like our single cell images and our expression data? Maybe you have other kinds of data modalities. Doesn't really matter here. And we would like to be able to translate between these very different data modalities. And in this case, unfortunately, as I said at the beginning of the talk, often we can actually not measure the same modalities in the same cell because they're both destructive to cells. So if I say an image, I will not get also the expression profile and the other way around as well. Well, what you can do is so we have two autoencoders. So one autoencoder, it goes from images to a latent space back to images. And for expression, I also go from expression, so single cell expression, to latent space back to single cell expression. And now, of course, if you take a population of cells, say I take all skin cells, and I at random take out some for imaging and at random take out some for sequencing, well, then it's the same population of cells. So in the latent space, they should match. Okay, so what you can do is they're just different views onto the same underlying biological phenomena, right? So in the latent space, these different distributions should actually match. And so there are different ways of doing this or of enforcing this machine learning, right? For example, you just have some classifier in the latent space that should not be able to tell whether, a particular cell comes from imaging land or comes from sequencing land. So that's a way of making the two distributions match in the latent space. Now, I think that this is also really interesting from a causal perspective. And let me just try to explain what this high-level idea is and why I think that something like this could actually help to identify or infer causal variables. So here is it written out. So I feel that, and there is now quite a lot of theory about, invariances and how to use invariances for getting at causal, not a causal representations, like causal relationships. But here I'm just proposing that, these kinds of latent spaces that we're actually getting when we're integrating different data modalities have a higher chance of actually being causal. Because causal features or underlying mechanisms should be invariant to how you're looking at it, right? If it is something intrinsically that is causal inside the cell, well, then it shouldn't matter whether I'm looking at it from imaging land or from expression land or from ataxic land or whatever land you want to look at it. It should always be there, right? It should be something that is preserved. So in fact, if you have different data modalities, like getting at these integrated latent spaces could also help you with learning causal features, just because they should be invariant to however I'm looking at these different kinds of modalities. So that's kind of the causal lens of, this kind of picture here of why different data modalities might actually be able to give you something or learn something about the causal features. And of course, it will really help, right, when you have something like images here, where, otherwise there is really no way of saying anything causal in terms of these images. Okay, so that's at a very high level, like one idea. And we're doing this quite a lot in terms of integrating different modalities. I think there's a lot of theory to be done that is not done here in terms of, how does this really give rise to causal features, etc. Something that we're very excited about looking at. Okay, so let me go one step further. And now it comes a simpler idea. Maybe, and many of you have probably seen this, and I like this picture very much by Judea Pearl, in this book, The Book of Why, where he has this really nice representation of thinking of different kinds of causal hierarchies, where, at the bottom here, you have just associations or correlations, right? Where, you answer questions like, how is this particular symptom related to a particular disease? So what does a symptom tell me about a disease, etc. Then all the way at the top is where you have counterfactuals. So this is, in fact, these are the kinds of questions that you can answer when you're learning your causal structural equation models. You're all the way here at the top. Okay, so these are questions like that you could, that are of the form, what would have happened had I not taken the aspirin? Right. So now, now that I've taken the aspirin, my headache is gone. But what would have happened had I not taken it? Well, I cannot go back and not take it, right? I'm always choosing one or the other. So that's why this is a counterfactual question. But these are exactly the kinds of, so if you learn a causal structural equation model, you're in fact able to answer these types of questions. Okay. That's what a causal structural equation model is done to do. So everything in causality, basically all of the research in causality is always here at this top level. Okay, this is where Julia Pearl puts humans, right? They are all the way here at the top. This is what they are, good at doing, and this is what causal structural equation models have been developed to do, to really answer these types of questions. However, Julia Pearl, he has like a middle layer here, and I want to propose that this one is in fact the one that I'm most excited about. And, I'm happy to be here at this Neanderthal level where interventions are sitting. And I think this level here is what is actually critical for a lot of biological questions. So here, what are the questions that you are trying to answer here? The questions are, well, if I take the aspirin, will my headache go away? So this is it's a causal prediction question, right? It's a question about an intervention that I'm just asking, well, if I take it, right, will it go away? This is a prediction problem because I can actually test it afterwards, right? I can then take it and then verify whether my headache actually went away and how I'm feeling about it. It's a very different problem and much easier than this problem up here. But I feel in science, right, we usually care about questions that I can actually verify afterwards. And so if I want to know something about what this knockout will do, right, I'm trying to predict what this drug will do. Well, I'm then going to go and afterwards verify it anyways, right? So you'll want to maybe, for example, do a virtual drug screen where you're ranking drugs, but then, your top hits, you're going to go and verify them. So these are the questions that I think you care about often in the biomedical sciences. And so I want to just, put in a point that maybe these questions are much easier than going all the way here to the top where you really have to learn your causal graphs. Maybe we can go and directly answer these types of questions here. And so this is the second idea, I don't know what it's called, idea one, but idea two of availability of interventional data could allow us to turn causal questions into actual prediction problems. So somehow make these questions easier. So we're not all the way here at the bottom of associations because we're trying to predict the effect of a perturbation, right? That's different than just trying to predict, say some conditional distribution, say, or something like that. And here's just an example of where I think, this question here is really the critical one that we try to answer is exactly that question that I told you about with SARS-CoV-2. So say here are all the different cancer cell lines. These are all different kinds of drugs and genetic perturbations. Black here means that this data point has been obtained. So in this case, it's expression data, but only 1,000 genes have been measured. So this is really a tensor where every dot here is a 1,000 dimensional vector. White means this data has not been obtained. And so you would really like to be able to predict what this particular drug will do in this particular cancer context, right? Or I could add a new column here, and I could add SARS-CoV-2 infected lung epithelial cells. And I would really love to know what all these drugs here that have been applied to some cell types will do in this new disease context. Okay, so that's this causal transportability problem that I mentioned all the way at the beginning. That's really falling into this landscape here, right? Also that the problem of predicting the effect of new interventions, if I want to just add here a new intervention, say a new molecule that hasn't been tested on anything yet, that also falls into this landscape here. So really, there is a distinction between these two levels. And I think it is interesting to also concentrate, and there is very little work that actually just concentrates on this level here. And so there is hope, at least, that we don't have to go, that we don't actually need these causal structural equation models and this full understanding of all of the causal relationships, because we're not doing anything at this level up here. Okay. So let me go one step further, third idea of how do I actually get around and do something like this? Okay, so I haven't even told you anything about how we actually go about solving some of these problems, just at a high level, how I'm thinking about these problems. So how would I go about solving some or attacking one of these problems? Maybe not solving. So if you come from a machine learning perspective, then I think it is quite natural to think of it in this way as a style transfer problem. So I'm sure you've all seen these nice pictures of Yosemite in the winter and Yosemite in the summer, how, this is just a generated picture of the same Yosemite picture, but just, added snow or removed snow, basically. And so I'm thinking of it in the same way. Okay, so now it's just not going from winter to summer, but just adding a drug. So say you have some cell, say here, cancer cell line. And, in the data set that we've just seen here, I actually know what happens when you apply this drug. Okay, so this is this cancer cell line. This is one drug. And I know what happens. And I also get to observe this over here. Okay, so this is before taking the drug, after taking the drug on this particular cancer cell line. Well, now let me look at a different, say, SARS-CoV-2 infected lung epithelial cells. Again, I get to see how they look when they're diseased, right? This is what you get to measure, this data set. This data was actually available very early on in the pandemic. So I know how these cells look like when they are infected by SARS-CoV-2. Well, now, wouldn't it be great, right, if I could just take like how style transfer works, right? So I embed these 3 data sets into a latent space. And this vector here corresponds to going from winter to summer, say, or in this case, applying a drug. Well, wouldn't it be great if I could just, take this vector, move it down here, right? I'm predicting that this is how this SARS-CoV-2 infected lung epithelial cells will look after applying the drug. And then, I decode it back out to my original space. And this is going to be my prediction of how this gene expression profile will look like. Well, this works very well on images, right? It works really well for adding hats to people, for adding like beards to people, etc. So all of these effects are, adding a hat, removing a hat, adding sunglasses, etc. So here I'm just thinking of it, and also it works really well in word embeddings, right? These kinds of these vector moving around different vectors. So maybe it could also work in drugs. Now, this is not something new and has been, in fact, if we talk to pharma industry, they have tried these kinds of things of using your standard autoencoders in the to actually move around the effect of drugs and predict this way in the latent space. So in fact, it doesn't work so well. And you can just test it, right? I can just take here one drug, take 2 different cell types or cell lines where this particular drug has been applied and just look at whether the effect of the drug in the latent space is aligned or not. So is it more aligned in the latent space as compared to the original space? That's a very simple just test that you can do, right? Because this moving around the vector only works if the effect of the drug in different cell types is in fact pointing in the same direction. Only then will this work, or is aligned in the latent space. And so, and here is again a very simple idea. And here I will actually give you a little bit of math. But it's a very simple idea. So autoencoders are usually used in this way, right? So autoencoders are usually used for going from a high dimensional space to a lower dimensional space and then back out to a high dimensional space. That's why I explained it at the beginning. Now here, what we're going to do is we're actually going to do something that might sound very crazy at the beginning. So we're going to go from our, whatever high dimensional space to an even larger dimensional space, and then back down to a lower dimensional, back down to our original dimensional space. Okay, so instead of using autoencoders for dimension reduction, which is, how they've always been used, we're going to use autoencoders for actually embedding into a larger dimensional space. Okay, so now why would this make sense in particular for this problem here? Well, if you want to find embeddings which are still, where you're still able to completely reconstruct your data so that you're not losing any information in your data set, but where you are in fact aligning certain directions, right? Here, I need to align the effect of a drug in order to be able to translate between them in different contexts or in different cell types. Well, it makes sense to go to a higher dimensional space because I have more directions in which I can actually align the effect of these different kinds of drugs, right? So just from that perspective, so going to a lower dimensional space, if you had linear things before, then now you're even squishing them up even more when you're going to a lower dimensional space as compared to when you're going to a higher dimensional space. Now you can, and in fact, if you just do PCA or something, you go to a very low dimensional space. In fact, you will also find very well aligned signatures, but you can get alignment by just throwing out basically all of the information. That also works, but that's probably not what you want to do, right? So you want to still actually have, all of your signal in the data afterwards, so that you can understand something about how this drug is working. So not just aligning things by just throwing out all of the information and just keeping, say one direction. and then things are also going to be aligned. But if you do this in a high dimensional space, you're actually not losing any information. I can in fact completely reconstruct the data set, but I can get alignment, and we can also prove that this actually gives rise to alignment. So I need to talk, and I want to talk a little bit about how this works and why this works on the theoretical side, and that's what I will do in the last 10 minutes or so that I still have, 5-7 minutes. But this is just at a very high level, this is the idea, okay? So that these overparameterized autoencoders, they provide more space to align causal effects. And so we really do need to, and they actually do work. So we can, they do work for this particular application. That's all I can say so far, right? So they do work for aligning these causal effects because we can just test it, right? We can test it on this data set and see whether it is better aligned in an overparameterized latent space as compared to an underparameterized one or in the original space. And you get, I can actually, the more you make your overparameterization bigger, the more in fact the more aligned your causal effects become. And so I think we do need to study their inductive biases and their interplay with causality, much more thoroughly than what I'm going to do in the next slide. But this is just more motivation for what you can actually do with it. And just to finish this, the story about drug repurposing. Well, if I can predict the effect of all of the drugs in the setting of SARS-CoV-2, well, then, if I can predict all of these effects of all these different drugs, well, then I can rank my drugs, right, according to which drug is most anti-correlated with the effect of a disease, and just rank my drugs. And then these would be my putative drugs that would actually work the best in this new disease context. And, there are some drugs here that at least some of these targets that we have here that are in fact in clinical trials. So, but what I want to do more here is actually tell you a bit, since this is a Foundations in ML lecture series, I want to tell you more about, just the theoretical understanding of why these overparameterized autoencoders should work, or why they don't even just learn the identity map, right? Because they have enough parameters to just learn the identity. If I'm going from a space in RD to something much larger dimensional and then back to RD, I do have enough parameters to just learn the identity map through it. But obviously it doesn't, because otherwise it would learn here, I would just get the same embedding as in my original space. So it certainly didn't learn the identity map. So a bit of theory of why is it that these autoencoders actually don't learn the identity map and might be learning something that is very interesting from a theoretical perspective. So I'm hoping that my pen will work. Okay, so, let's just think about it very briefly. So we have, say, here are our N samples, training examples, and they live in RK. And the number of samples here is smaller than the dimension. It's one way, or you can also just think of the latent space as much larger. And so this is what an autoencoder does, right? So this is my training examples. Psi is my autoencoder map, which goes from RK to RK. So this is what comes out of the autoencoder. This is what I'm putting into the autoencoder, and I'm trying to minimize reconstruction error. So let's do the most extreme setting, and just to get a little bit of intuition, and let's just say I only have one training example. Okay, so one training example, and let's say something very extreme where, say, this map is the simplest I can possibly think of, so it's just linear. So then, what I'm trying to do is just minimize this, right? Where, A is in RK times K. And, this is just a vector, right, in RK. Okay, so this would be this problem here if my autoencoder is just linear and I just have one training example, so the most extreme setting. So, so there are many solutions to this problem, right? So first of all, I can always train this to get zero training error. But in terms of my matrices A here, there are so many different solutions. It could be the identity, right? That's one solution. Yes, it could be the identity matrix. But it could also be a rank 1 matrix, which just projects everything to X, right? Or it could be a rank 2 matrix, rank 3 matrix, etc. So there are many solutions to this particular problem, as there are many solutions here in the nonlinear setting to this particular problem. So the question is really, what does an autoencoder learn, right? And what we saw before in the examples is that on that data set is that it doesn't learn the identity map. So it doesn't here learn the identity map. And in fact, we also know for this very simple problem here, this has been proven like a long time ago, that, if you just, so here it depends on the initialization, right? So if you initialize like neural networks do, this weight matrix here close to 0, then the solution that is actually coming out is the rank 1 matrix. Okay, so this would just be the projection onto X. So the question is that also the case in the nonlinear setting? And you can actually train your neural network or here an autoencoder and just do it on one training example. And what you'll see what comes out is actually something that basically looks like the point map. Okay, so here I trained it on these, Adit, this is work by my PhD student Adit and together with Misha Belkin at UC San Diego. So if you train this just on one particular image, right, then you feed in all kinds of training images, then you'll see that what actually comes out is always a training image itself. So this is a point map that was learned, not just a rank 1 map, but actually a point map, which is much more extreme because you never see the negative of these bunnies, for example. Now, often, we would get a reply that, well, this is what I would have expected. Well, maybe it shouldn't be what you have expected because, if you actually use a less deep network, you can in fact get, and so these UNet autoencoders are in fact quite deep. So if you use something less deep, you can actually get something that is very far away from the identity map, very far away from the point map, namely something that basically looks like the identity map. Okay, again, trained on one image. Here, these are my test examples, and you see that what comes out looks actually pretty similar to what you put in. And if you do a linear autoencoder, then in fact, it's very easy to prove that you actually always get linear combinations of training examples. So, there is something interesting going on, right? And so the question is, and, I will just want to just get to the punchline, so I have some time for questions. The question is, what is the map that is actually being learned here? And, let me just, in terms of intuition for the theoretical result that we have, what is really nice about an autoencoder is that it is a map from RD to RD. So what you can do is like whatever comes out here of your autoencoder, you can put it back into the autoencoder. Just put it back in as a new test example and see what happens if you just iterate the map. And, if this is really the identity map that was learned here, then, this horse should always come out as a horse. But what is interesting, and this is exactly what we proved, is that, in fact, this is not what it's learning here, although there are some papers that would suggest this is these autoencoders, these overparameterized ones, they do not learn the identity map. They cannot. In fact, what they're learning are maps that are contractive at the training examples. So if you do this and you just iterate your map, then what will come out is in fact one of your training examples. And this is here just shown on something that was trained on 500 examples. You see that, sometimes here I'm putting in a training example with a whole lot of noise on top of it. If I iterate it, you can converge to a different training example, right? I'm not saying anything about the regions of attraction, but what is actually happening is that you do learn maps that are contractive at the training example. So yes, they might look like maps that are learning something like the identity map at the beginning. They're not. They might look like as extreme as here if you go very deep, where they're so attractive that already in one iteration you're actually ending up in your training example. That's what is really happening here. It's just so attractive that already in one iteration I'm actually at the training example. So that's what that does to you. And so we do have a proof now that, so this was a proof in PNAS where we just had it for one training example. Now we do have the general proof that, these overparameterized autoencoders, they in fact will every one of your training examples, if you just make it wide enough and deep enough, your autoencoder, it will always have all of your training examples be attractive fixed points. So that's quite interesting because it tells you that the maps that are learned by these overparameterized autoencoders are interesting maps, right? They are in fact self-regularizing. So they do lead to more and more alignment, or at least they're making things more points more similar to each other. And in fact, also directions, like what we have seen before for causal effects more similar to each other. And so I think it is an important open problem of like, how much of this, can we make this more theoretical? That, now we know some alignment properties of these overparameterized autoencoders. Can we really connect it, as we've seen it happen, work quite well on these causal questions of predicting the effect of new perturbations or perturbations across different contexts? Can we make this more theoretical and really understand the inductive biases of these overparameterized autoencoders and how they're related to causal transportability? But generally, I think these autoencoders, there is a really nice intersection that we have to understand better between causality and representation learning. Here for us, representation learning always done through autoencoders just because we can actually prove things about them and really understand them a little bit better. But of course, there are similar questions also for GANs, etc., for all kinds of different representation learning methods. Okay, so a lot of open questions in terms of, what are these areas? How do these these areas of attraction actually look like, etc. And how is really this relationship to causality, like just to understand all these inductive biases a bit better. So with this, let me end and just thank, and I already pointed this out. So a lot of this work at the end was by my PhD student, Odit, with my collaborator, Misha Belkin, and everything on the biological side was in collaboration with G.V. Shivashankar's lab at ETH Zurich. And with this, let me end and thank you all, and of course also thank funding. And happy to take any more questions.
Thanks, Caroline. That was a really interesting talk. Yeah, we have some time for questions. So if anyone wants to raise their hand or type in the chat window, I think we can take a few questions. Okay, I have a question if no one else wants to raise their hand. I missed the connection between some of the transportability questions that you were talking about earlier in the talks, transporting the effect of the drug with that last bit about theory. And I think it went a little fast for me to keep up with. So would you mind just kind of, explaining that a little bit?
Yeah, of course. Yes. So, okay. So, and I think, you didn't miss it. It's just that we don't have the full theory of why is it actually really working, right? So for now, what we have is that, so we do see that, we can see in this particular data set that it does align the effect of drugs across different cell types that you can just test. You have a data set and you can actually, you have it, and you have drugs applied to different cell types. And so we can see that it is more aligned if you're using over-parameterized autoencoders as compared to the standard autoencoders, and this has also been seen in pharma industry. So it doesn't actually work in pharma industry. So they're excited that this seems to work in the over-parameterized setting. So then this motivated us to look into what these autoencoders actually learn. So the intuition was really that we have more space to align the different directions. But then, then we have to learn, well, first of all, why doesn't it even learn the identity map, since it could learn the identity map? So that's when we saw that it learns these contractive maps at the training examples, right? So it is self-regularizing, so that's why it doesn't learn the identity map. So the question is, how is this? And our intuition was wrong. So first we thought that how this actually works is because what we proved is that, right, if you have training, so that zero-dimensional points will become basically more aligned. So if you have your training examples, you're becoming more similar to one of your training examples. That's what these maps do. We were thinking that maybe this also holds for one-dimensional manifold. So that was really the intuition. So here I have a one-dimensional manifold, like one direction, basically. Mm-hmm. Well, is it a case that it's just holding also for one-dimensional things, and then maybe even for two-dimensional things, just for low-dimensional manifolds? Maybe this is still correct, but this is not, I think, in terms of math, how we have now proven things in more recent work, is that it's actually stretching. It's just using these additional dimensions to stretch out data. And in particular, that's how it is aligning these directions. So if you have additional, dimensions where you can stretch things out, then of course, this is a way of you actually being able to align causal effects in dimensions that you're not using before, therefore helpful, therefore you can still reconstruct fully and don't get any hit, and can, very nicely transport between different cell types.
Okay, interesting. And you're not changing the training objective at all?
Nothing.
Like that.
No.
It's just,
yeah.
Okay.
Yeah.
Cool. I think we have a question in the chat window here from Joseph. Maybe I'll read it aloud just so everyone is on the same page. So can association studies still help as a first step before moving on to studying causality within a system? Or is perturbation data very easy to obtain or extend to unknown domains without first knowing the associations?
Yeah, I think these are really great questions. So I think we're just in a lucky position in the biomedical sciences that you actually do have a lot of interventional data. So if you, I think just based on the initial work that I showed, like if you just have observational data, I mean, it's just, we know it's a very hard problem, right? And so you just need huge amounts of samples in order to be able to say anything about the underlying causal relationships. And of course, we also know that not all causal relations can be identified. That's another problem as well. So for that, you also need perturbational data. So there are two problems in terms of identifiability of the causal directions, and then in addition, in terms of the sample sizes that you actually need. But of course, you do whatever you can, right? If you don't have perturbations, you just have to know the limitations. Yeah.
And do you have time for one more question then from David?
Yeah, I'm happy to.
Hi, thanks. I enjoyed your talk
David.
very much. I have a question about your last answer to the question. I thought you were both learning to, in the autoencoders, you're both learning to reconstruct it, but you're also simultaneously trying to learn the alignment.
Correct.
Is that correct? Okay, because I thought your previous answer was saying that you're only reconstructing. Okay, so your story over here.
Let me
your story
just
over here. Yes.
No, go ahead. Sorry.
Oh, so your story here is this one's here is more than just your images are sort of very much oversimplifying it because you are learning the alignment as well. Okay.
That's correct. So here is the data. So how we're testing it is that, you know, yes, in the overparameterized setting, you're not losing any information. So you have like perfect reconstruction. Of course, as we saw, you can always train this to zero training error. But in addition, you also get just for free the alignment of these different drugs across different contexts. But
for free means you're learning them from the data.
No. I'm not adding any constraints that they have to be aligned. So that's the nice thing.
So you're not sort of adding any constraints that say here you have some alignments and now you're trying to learn.
Nope.
To learn them.
It's like in the word-to-vec embedding, right? You're not adding constraints to make these things align.
Okay.
Yeah. So that's a nice thing. So yes, so because you don't know that right beforehand. I mean, you could, if you have some training examples, etc., you could add in some constraints, but you actually don't need it. That's the nice thing. You don't need it, and it's very similar to word2vec, that you actually just get this out for free. And I think it is an important question to understand: when do you get alignment and when do you not? And you know, I'm happy. So basically, the additional directions that you're going to use are the principal components of your data set. So this whole thing can only work when your top principal components are not pointing in the direction of your drugs. When basically the top principal components in the data set are something like different cell types, which is basically orthogonal to the direction of the drugs, because you're just adding these directions in order to align things, you know, and so you don't want to have confounding between, you know, alignment that comes from your top principle, from the additional directions, and the alignment that you actually want in your drugs. So yeah, so it's really important to understand better, like in which direction are these data points going to be stretched. Because if you're suddenly just stretching into the direction of a particular drug, then, you know, everything will be aligned with that one drug, and that's not what you want.
Thank you.
Yeah.
Is it okay if I ask one more question then to follow up on that? Is there any, I mean, have you thought at all about the sort of induced geometry in the latent space? I mean, do we have any real reason to believe that, you know, alignment of vectors is what is meaningful in this context, or I guess in any context where you're using these type of systems?
Yeah, so there are multiple questions here. So one about alignment. So yeah, so okay, so we would want something. So first of all, why is it that we want this kind of geometry? Is because we really want to solve this problem, right? We want to somehow solve this problem of predicting the effect of perturbations across different contexts and predicting the effect of new perturbations. So I would like to have a latent space, you know, in terms of future work. If you have like in terms of new perturbations, right? You would like to have, if you do combinations of perturbations, to be able to add up one perturbation after the other in the latent space, because otherwise I'll never be able to predict the effect of combinations. So you want some latent space that will become hopefully something like linear and hopefully something like additive. Otherwise, there's just very little hope on doing anything. But yeah, if you can think of other geometries where something like this might be possible, that you know, but you want something simple. So that's the first thing about what you would like it to be. Now, in terms of what is learned with different like non-linearities, etc., right? That's all open. I mean, here is just like even the geometry of these attractor landscapes really depends on which non-linearity you're using. So this is just here like 4 training examples in R2 and just showing you, you know, all of these points around here will be contracting to this training example, etc. So we have no proofs on how these attractor landscapes look like, what would be better non-linearities to use, etc., and how do these change. So none of this we know. Yeah.
Great, thanks a lot. Okay, well, I guess if we have no more questions, thank you very much. Let's thank Caroline one more time for this very interesting talk, and thanks very much for joining us.
Thank you very much.